Loud. Whoops.
Oh, now I could hear you now. I don't know what's wrong here.
Okay, all right.
Should I try to share the slide?
Yep, let's see how that works. Make sure it works.
Okay, hold on.
Do I need to give you access? No, I guess not.
I guess you don't. Do you see it?
Yep, but I'm looking at, I don't understand why I'm looking at my screen here. Turn that off. Okay. Turn this off. Okay. I can't see it now. Yinjie Qiu. So Yinjie obtained his bachelor's degree in agronomy from Sichuan Agriculture University in China. He did his PhD or his master's degree at South Dakota State with Yang Yan, and he worked on Fusarium head blight of wheat as well as perennial maize. And then he conducted his PhD with Eric Watkins here at the University of Minnesota in the APS program on turfgrass genomics. And currently he's a CFAN's Diversity Scholars postdoctoral fellow working with Candy Hirsch on Maize genomics. And today he's going to talk about, I think, a lot about transposable elements and their impact on genomic variation for crop improvement. So I'll let him take over and tell us what he's been working on.
Cool. Thank you, Gary, for the introduction. So hello, everyone. I've been at the U of M for quite some time since I started the PhD program here. So I feel like I'm somewhat like an old dinosaur here. So if you're new to this program, I'd like to tell a little bit more about myself. So I was born and raised in Chengdu, Sichuan, China. So it's like in the heart of China, and the city is huge, lots of people, and also we have lots of good food, hot pots, and that's also the home state of the panda bears. So as you can see, there is a panda breeding center just outside of suburbs of the city. And also it has lots of cultural diversity where there's like the Sichuan opera is quite famous around the world. So like Gary said, I started my PhD research here with a turfgrass group where I look at a grass called fine fescue. Throughout my PhD study, I realized there are so many different diversities in terms of like inter or intra differences between those grasses. And also we observed a substantial amount of DNA content variation. And after graduation, I am working with Candy Hirsch on the maize genomics, where maize is also a species that has lots of diversity. And that has been my passion to look into them. So plant diversity comes in all different types and form. It could be underground, where we can see the root architecture is different from genotypes to genotype. And we can also see above ground tissue, like the plant height is a great example in this picture showing here. On the left, I can easily, the genotype on the left can easily reach its tassel. And the genotype on the right, I just cannot even get in close to there. I probably need a ladder. Even like the tallest person in our group, Claire Menard, probably cannot even reach it. So that's just another example about interspecies diversity. You might think it's far away from you, but it's actually not. There are also examples in horticultural crops, such as tomato, where we see the fruit in terms of its size or color, and flower size differences, and leaf phenotypes, and also the clusters, the number cluster for tomato plant, where it matters a lot for the yield. And for flowers, if you're a person like me who loves orchids, I do see a lot of different species, different phenotypes within one species, but also doing that, you are posing a risk on yourself where you're ready to turn your house into a jungle. So moving forward, there are multiple ways we can evaluate the genotype to see the differences between them. We can start with the cytogenetics approach to use the dye to stain the chromosome and see the differences, the karyotype. And we also have tools to use the molecular markers to do the genotyping for the plants to see their differences just in terms of DNAs. And eventually we're at the stage where we're able to generate a lot of sequencing data, and we'll be able to look at the single nucleotide polymorphism, where we're able to look at the plant diversity at further depths. So from those single nucleotide polymorphism marker to either quantitative trait locus mapping or genome-wide association study, it's heavily based on linkage, this equilibrium, where it's a non-random association of alleles at the different loci of a given population. So one example here, if the genetic variant is highlighted in red here, and we have a SNP close to it, where when you conduct the genome-wide association study, you will be able to find a candidate hit. But when the SNP is nowhere close to the variant, which we frame it that's in low LD with the variant, we will not be able to capture the signal. However, that does not mean this variant is not there. In the practical example, let's walk through this together. If we want to look for a plant that is dwarf, so what we could do is to do lots of resequencing or genotyping by sequencing, where we can generate a lot of single nucleotide polymorphism markers, showing here A and G. But also from resequencing, we can see some other genetic characteristics that cannot be described as SNPs. Let's don't think about them for now and move forward just with a single nucleotide polymorphism markers. So here, when you use those markers to conduct a genome-wide association study, where you will be able to see some signal that the SNP is linked to the causing variant of the phenotype you are seeing. But obviously, we don't have information for the variants that's not SNP in this case. So through this approach, lots of traits could be analyzed through the association study, where we can see that it has been used in maize and also wheat, and example of land races about their cold tolerance in rice, and ornamental crops or a horticultural crop like tomato will be able to find genetic locus associated with its flavor. But meanwhile, we do indeed discover lots of association from using SNP markers, but there are also traits out there that cannot be found variants, like why it's different. For example, like kernel carotenoid concentration, or the fruit size in tomato, which is causing by copy number variation. And here's another example is soybean nematode resistance, where it cannot be located just by SNP marker. The final example I'm going to show here is the sorghum seed color, which is also not being found through a single nucleotide polymorphism markers. However, they will be able to locate it through another type of genetic characters, which hasn't been touched that often before. It's structural variant. So there are three types of structural variation, either presence absence variation, copy number variation, or a larger scale of variation like chromosomal inversion or translocation. So let's keep that in mind and revisit the figure we're seeing here, where those variants are not SNPs. Now we know they are structural variants. And it is not easy to identify structural variants when you only have one reference genome. Here's a place we brought in the concept of pan-genomics, where you have multiple reference genomes and you will be able to conduct comparative study to see what is different. And you will be able to characterize those structural variants such as insertion or deletion. So doing the same thing, when you treat an insertion deletion, those structural variant just next SNPs, you'll likely be able to find more locus associated with the trait you're looking at. So downstream, it's the same, where we can use this knowledge for the crop improvement, either through molecular marker assisted breeding or genomic selection, or for genetics or gene editing. So at this stage, we no longer just have a karyotype or marker phenotyping, SNP phenotyping. We will also have the structural variant based genotyping. So now we know the possible power, how the pan-genomics is going to bring for plant breeding and how important those structural variants are that we have not looked at before. So here's more example about the pan-genomics and structural variants for crop improvement. In soybean, where the pan-genomics, through pan-genomics, we're able to discover locusts that associate with the trait luster, seed luster. And for tomato structural variants, we know it's part of the fruit size. The reason why the fruit size is different. And for oilseed, through the presence-absence variation, where we're able to find out genetic locus conform to the seed architecture. For understudied crops or non-model species such as sunflowers, where the pan-genomics approach allow us to look into the crop domestication and also gene introgression for disease resistance such as powdery mildew. And for peach, where structural variants identified in the genome confirm the day to maturation in this crop, which has not been found before just using SNPs. And final example I want to show here is about the pepper pan-genome, where a gene presence-absence variation confirmed the red carotenoids content variation in the species. And very recently, our own Department of Horticulture Science potato research group participated in a study where they look at the tetraploid potato pan-genomics, where they are able to discover structural variation or embedded deleterious allele and dysfunctional allele in potato. And those information could be very important for the breeders for the future breeding and genetics work. So, I work with corn group. What about corn? Maize. So obviously there's a lot of good things about corn, where it has lots of phenotypic diversity in terms of the ear polymorphism or plant height and some other traits. But also it has another complication where it has undergone whole genome duplication and returned to diploid. So pretty much when you search corn genome against the sorghum genome, you will be able to identify maize actually have two subgenomes, M1 and M2. And then the bad part about it is has a large genome size with more than 85% of the genome is redundant sequence, which include large amount of transposable element. For this case, it makes the genome assembly very challenging. And last but not least, maize has around 40,000 genes, which is quite a lot. So taking all those factors into consideration, when we recap the maize pan-genomics research, we started with, by looking at transcriptome due to the large genome size of maize, where more than 500 genotypes were looked at one time to look at the gene content variation. And recently, European maize genomics research, where they sequenced 4 more genomes, and they were able to look at some intra-species variation and also their gene content diversity. So in 2021, we participated in research to sequence the maize nest association mapping panel, where it includes 26 diverse maize lines. So when we look at the background of those 26 lines, as you can see, they're selected highly diverse from highly diverse backgrounds. They include like non-stiff stock, tropical, subtropical plants, popcorn, mixed germplasm, and sweet corn, and stiff stock. So just by looking at the ear of the name panel, where we already see a substantial amount of variation just by the first look. And after the genome assembly, we were able to look at the genome size across all the 26 name lines. They're undergone the same process for the assembly, so the data will be highly comparable. As we can see, the genomes on the left, which is more like temperate lines, it's around 2,100 something there. While on the right-hand side, we can see all the tropical lines generally has a little bit bigger, large genome size. With a high quality reference genome out there, we were able to look further into the genetic architecture of the maize genome. For example, we see transposable element variation, where across the 26 name lines, we see each TE family has different proportion within the Maize genome. And we're able to see large insertion or deletion and knob type structure in the Maize genome, where we're able to conduct an association study to analyze if a squeeze explains some sort of phenotypic variation, such as disease, agronomic or morphological trait, or metabolic traits. Last but not least is our major interest. We want to look at the gene content variation. Now we know the fact that the Maize genome across the 26, they all have different genome size. What about the gene content? So after annotating the whole genome using both ab initio and evidence gene annotation, we're able to see the difference in gene number by itself, where there are some genotypes has like 40,000 genes, and also their genotype has 41,000 genes. So there are quite some differences. So when you put those numbers together, we wonder there are more than 1.5 million annotated Maize genes from the 26 genome. But how many of them are actually unique to each genotype, and how many are shared across different genotypes? I just don't believe all the 1 million genes are unique to themselves. So in order to answer this question, we carry out analyses of pan-gene identification, where we grab a gene from genotype 1 and we search it in all the 24 genomes. So what we can see here, when the gene is located at the same region with high sequence identity, we consider them as one pan-gene, where in this case, we have pan-gene 1 and it's present in genotype A, B, and D, while it's absent in genotype C. So you may wonder, they're gonna have a unique gene that's in one genotype, not in the other. So we have another step after that to look further, where we look at the gene in genotype B that's not in genotype A and try to search around to see if there's any other genotype that gene is present in. So in this case, we see the pan-gene 2 that's present in genotype B and C, while it's absent in genotype A and D. So first trivia question, out of the 1.05 million genes, how many of them are shared in the Maize genome? I just wish we could have some sorts of in-person interaction in this case, but on Zoom, it's just almost impossible. So I like to have you think about the number and keep it in your mind before we move forward. And if you get the number right, message me after the seminar. I'd be happy to send you a pretty corn that we grow out in the field. All right. After analysis, we found out there's 103,000 pan genes out of the 26 Maize genomes. To be able to look at the pan gene frequency, how many gene is which genes sharing how many genotypes, we're able to break these stats down into 3 classes. And we started to name them at the private gene, where the gene is only present in one genotype. And when the gene is present between 2 to 23 genotypes, we consider them as a dispensable gene. And for anywhere above, that includes 24, 25, 26, we consider those genes as core gene or near core gene. As you can recall from our previous introduction about the Maize subgenome, they all have the corresponding gene in sorghum. So you will think those genes should be in core gene, which is indeed the case. We see most M1 and M2 Maize subgenomes are located either at the core or near core gene. And we also see just the pan gene itself, there's a large number of genes, 27,000 genes that's shared across all 26 genotypes, while we do see a substantial amount of gene that's private to each genotype. So when we generate the stats for each pan gene class, where we see there's close to 50% of the gene that's considered as a dispensable part of the genome, and around 27% of gene is considered as a core genes. and close to 20% of the gene is private genes. And there's another way we can look at this is to see the gene content variation through this heat map, where the orange color we study over here is the core genes. And as the pan-gene frequency decrease, we're able to see more fluctuation in terms of consistency where the gene is present or gene cluster is present in the 26 name genomes. And we finished up with all those private genes here. So let's look at this pie chart. There are 20% of the genes are private genes, but how about we break them down by genotype? Well, the private genes still take up a large proportion of the genome by within itself. So what we can see here, it started with core gene where it's more than 60%, followed by near core gene. Then we see there's a large chunk of dispensable gene. In this case, after we dissect all those pan-gene distribution by its genotype, we see there's only a very minimum amount of private gene across its genomes. The number is somewhere above 1,000. So the next feature, we look at those pan-genes about its phylostrata. So as I mentioned earlier, maize and sorghum, they are really close to each other and believed to come from the same grass long time ago. So we will think whatever gene is essential for the plant development should be in the pan-gene class, core gene class. Let's see if that's really following our hypotheses. So when we look at the phylostrata of all those pan-genes, we can see for core gene, there's a very small portion of gene that's just maize specific, and all the rest belongs to the higher phylostrata. As the pan-gene frequency decrease, which we will think it's getting more maize specific, and our data is also suggesting that. So that's another feature we look at. The next part, we all know tandem duplication create lots of transcript abundance variation, which eventually leads to the trait phenotype differences we're seeing. So for all those pan-gene, there's 16,000 pan-gene that were tandemly duplicated. When you break them apart into the private tandem duplicates, on average, each genome has around 270 of them. That's just unique to itself. And those features has not been looked into before, which could possibly explain a lot of phenotypic variation we're seeing, just like example showing the potato research where copy number variation matters about the fruit weight. So we know all the genes, but if they don't express, how do we know their features? So the next question we try to look into is to see how many tissue type each pan-gene expressed in. And also, not just about yes or no type of expression, we also want to know the expression level of the gene. To do that, we did RNA sequencing on 8 tissue types, where it includes shoot, root, and the base of the leaf, middle part of the leaf, and the tip of the leaf. Beyond that, we also look at the reproductive tissues such as tassel, and anther, and also the immature cup. There are lots of genotypes also have information about embryo and endosperm, but because tropical lines, it's not gonna flower in Minnesota because it gets cold in September or October, we're not able to capture those tissue. So moving forward, we're just looking at the 8 tissue types that's consistent across the 26 genomes. So first, let's look at the expression by tissue type. So on the top, we have the pan-gene class where we have core gene, near core gene, dispensable gene, and private genes. And on the bottom, we have the color-coded different shade for tissue types, starting from 0 tissue types to all the way 8 tissue types. So we will think for core gene, they should express in more tissue type because they are likely to be a hop gene. Is that really the case based on our data? So when we look at this, there's close to half of the core gene that has expression in 8 genotypes. As we moving forward for near core gene, where the frequency, the pan-gene frequency decrease a little bit, and we also see the number of tissue type the gene expressed in is decreasing. Eventually, we drop to the dispensable gene where we can see there's only a small proportion where that gene expressed in A tissue types. And finally, we're looking at the private genes where you can obviously see the number of private gene is substantially lower than any of the other pan-gene class. And also noted that the private gene in the 26 named genomes might not necessarily be private for like Maize in the diversity panel. So that's why you're seeing there's a little discrepancy based on the trends we're looking at. Besides how many tissue types it's expressed, we also look at the expression level where the core gene is having a higher expression level than any other pan-gene class. Again, for private genes, there's a little bit difference, but just because it might not be really private genes in the larger Maize panel. So now we think 100,000 genes is a lot of gene. Does that capture the whole diversity of the Maize? Well, obviously, I noted already there are some private genes and not private because there's still gene out there. So the European Maize pan-genome study where they look at 6 genomes and you can see the gene content in orange color, it's far from saturation. When we did the same thing to our 26 named Maize genomes, where we see the substantial increase at the beginning where we're able to capture a lot of gene content variation across different lines. And you can also see the speed of increase, it's starting to slowing down. So we're going somewhere, but we're not there yet. And also between the temperate and tropical lines, you can see the intergroup variation of gene content is a little bit smaller than pulling everything together, which suggests there's also gene content variation just across different heterotic groups. So, all right, we have the named pan gene out there. So there are more than 100,000 genes for the Maize genome. But when you look at each genome individually, there's 40,000. That means the total pan gene is almost like 2.5 of the single Maize genome, suggests the magnitude of the gene content diversity in Maize. And also there are so many gene content variation like tandem duplication where we see there are lots of private tandem duplicates belong to one genome. So what's next? What I present here is just a tip of the iceberg of the pan-genomics in corn, in the named pan-genome, I should say. So further, we're going to characterize the nucleotide diversity between different pan-gene class and do some sort of functional characterization, and along with the gene co-expression network. So on February the 14th, the APS Seminar, the master student in our group, Amanda Gilbert, will present her proposal research and talk about those content in depth. So now we talk a lot about the named pan-genome. What if we go beyond the 26 genomes? So what we are working on right now is we have done the whole genome resequencing for 509 Maize genotype. We're able to characterize the pan structural variants of the whole panel, where we detected more than 250,000 deletion that's larger than 25 base pair long, which has not been noted before just by SNP markers. And when we see the saturation of the structural variant, we can see also at the very beginning, there is a fast increase capturing all the diversity. And eventually the speed is slowing down around 118 genotype. We're able to capture 90% of the total number of deletion we detected. And when we have close to 200 genotypes, we're able to see there's 95%. So we're going somewhere. And last but not least, we are also looking at the pan-genome, which is obviously the core of our interest. So from our assembly, we generate more than 1 million non-reference contacts, suggesting there's so many diversity out there in this diversity panel. And we are working on annotating all the contact, non-reference contact, not all the contacts is gonna have gene. So it's not gonna be like a million more genes. So stay tuned for the future updates when we get that piece together. While I cannot tell you how the Pan gene looks just in the 509 genotypes, but the next part, I'm gonna tell you some stories. We're using this data to look at the transposable element in this Maize diversity panel. So we're almost 25 minutes into the talk. I feel like everybody might get tired a little bit. So I'm gonna share something that's beyond the talk. So last Wednesday, I was asked if I'm interested in giving a seminar. I was like, great. That'd be fun to present some of our studies while I have limited amount of time to think about a title. So I asked our group, like, if someone could come up with a cool title that's fun, informative, I'm gonna buy that person a beer. So we come up with lots of titles, and I'm just gonna select two over here. One is tips and pans in the Maize Pan-Genomes. The second one is heavily towards the transposable element. Stop ignoring T's and add more genotypes to your references. A lesson learned from the Maize. When I brought those titles to Candy, she shoot it down, but she does not know the background that somebody's gonna get a beer for the title. So I guess now I owe Candy a beer. But well, since we're talking about T's, what is T anyway? I mentioned multiple times, but I didn't really give a specific example or concept for it. Transposable elements is also known as jumping gene. That's a DNA sequence that can change its position through cut and paste or copy and paste mechanism and move within the genome. And sometimes those T's gonna create lots of different phenotypes and also play a role in the genome size content differences. And for a long time, it's been considered as a junk DNA just because we don't know too much about it. And it's also been considered as a dark matter within the genome. However, is this dark matter really that far away from you? So I'm gonna show you a few examples you might be familiar with. So for strawberry, there's a T insertion that makes the strawberry lose its color, where we nowadays have those kind of albino type of strawberry. This is just eco-type, but in Japan, there's like eatable type that's much, much bigger. If you say you're not familiar with strawberries, how about those orchids in the store, either Home Depot, Target, Cobb, you name it. So T insertion also changed the color density of those orchids. Last but not least, there's a bioRxiv manuscript came out this year, 2021, where they hypothesized the T insertion actually make human lose our tail. So as shown in this figure where they did the transgenic mouse, where they are able to kind of remove the tail just by manipulating those transposable element insertion. I need to note that this paper is still under review. I have not given a deep critical thinking about it. So I'm just providing the fact out there that might be interesting to you. And we'll see if it holds true after the peer review. So I've given you some example that we can all relate it to. And there are also some classic example in Maize. So in 1949, this is actually how those jumping gene transposable elements were discovered. And over time, we're able to learn the T not just deal with the kernel the kernel color variation. We also know it manipulates the architecture of Maize, flowering time, and also the stiffness of the stalk. So the progress for the T insertional polymorphism, which is crucial to characterize those phenotypic diversity, started from one locus each time. Eventually, we're able to look at multiple locus each time because we have lots of reference genome. But at least the 2019, we only have 4. And until last year, like the name publication released where we're able to annotate 26 named genome at once, where we could hold the annotation consistent, accountable, and further answer some more questions about the T's. So there's a paper we're gonna release on bioRxiv soon that talk about how T is playing a role in manipulating the genome size between the tropical and temperate Maize genomes. Throughout this study, we're able to look at that pan-TE family, where we can see the number of TE family is getting saturation as we added close to 26 genomes. And we're also able to look at how each different TE families were driving the genome size differences across temperate and a tropical Maize line. So Shujun did a lot of work for this manuscript, and I'm very excited for the finding he get out of this. So when the paper goes online on bioRxiv, feel free to look it up. I think it's very cool. So that's 26 genomes. Can we do a association study just use the 26? That's going to be very challenging. So for the TE polymorphism at the population scale, those research has been done on couple crops or plants, such as tomato, rice, Arabidopsis, and they all develop tools to call the TE insertional polymorphism. But those tools is just suitable to deal with a small amount of TE, not like when the genome has 85% of the redundant sequences in there. And just because there's a large amount of TE Maize genome, those pipelines tend to create lots of false positive calls. So to solve the problem where we want to look at if we can use the machine learning model to use the data we have to predict the presence or absence of the TE information based on the references. So in order to develop a training set where we used for the resequencing data from B73, MO17, PH27, W22 for reference genomes, and we map them all to the all four reference genome. Where in the example, when we map W22 resequencing rates to B73, we're able to look at B73 specific TE, the B73 based TEs, and calculate the left coverage of the TE and right coverage. Besides that, we also have another piece of information where we know if the T is truly present or absent between those two genomes through the comparative genomic study by Anderson et al. In 2019. So now we have three pieces of information where we have left side of the coverage of the T, right side of the coverage of the T, and also whether this T is present or absent. So we try to visualize the data. So after we plot the left and right coverage on this graph, we can see there's like some clouds clustering here, and also there's another cloud clustering here. After we're filling the information about the T that's present or absent, it's quite cool to see that when one end of the coverage of the T is high, the other end is low, this T is likely to be absent. While when both ends are quite high, there is a good confidence probability where this T is truly present. So we use those information with right coverage, left coverage of the T and the T order to construct a random forest machine learning model, where this model will output the probability where this T is present. So obviously it's great to see the probability, but also we need to look at the accuracy of the random forest model. So in this example, where we map PH207 reads to all four reference genome, we're able to see that PH207 does not have those private gene from B73, while B73 also shares some Ts with the PH207. PH207 to itself, where we see there's very minimum amounts of T that's been called absent, which is great. So we have all this probability to be able to assess the accuracy. We need to generate metrics such as false positive, false negative, true positive, true negative, and also how many total T we're able to capture through this method. So at each probability, we plot all the metrics I just talked about, where we can start from the bottom about the false positive and false negative, where we see no matter what cutoff you're going to use, it's highly consistent across all of them. And when we look at the true positive, true negative, and also the total percentage of a T being captured through this method, where we can see there's, for the true positive, negative, it's also consistent, while for the total number of T we're able to capture is decreasing after 0.7. So here we use 0.7 as a probability cutoff where we define the T is present in this population. So now we have a method to call the T presence absence, and we're going to apply this method onto the 509 genotypes Wisconsin diversity panel I just talked about. And also I need to note that those lines are selected to flower within 2-week window in northern region. So we start with 509 genotypes. We map them to all the 4 reference genome, just like our built-in training set, where we're able to calculate the coverage for the left end and right end of the TE. And we plug into the random forest machine learning model. So we then know the presence absence call for more than 1 million annotated TEs. So before we move further, we want to see if those TE truly, if the TE presence absence call is truly accurate, which we compare the genetic distance generated by the SNP data versus the T presence absence call, where on the axis, we can see that it's a TE call, and x-axis is the SNP call. So you can see it's highly correlated. So that gave us the confidence, the quality of the call. And then we move forward, just like the pan genes, there are also pan Ts. There are Ts shared across different number of genomes. In this case here, we can see there are Ts shared between all 4 or 3 or even just 2 of the genome. So we wonder if the frequency of the T presence absence call is consistent across different genomes when they are shared Ts. And that will give us capacity if we could decide to merge those T frequency calls. So to do that, we are looking at the pairwise, the TE population frequency comparison. So for this zoomed up one, we can see the x-axis is the B73 reference genome, and on the y-axis is the frequency call based on MO17 reference genome. As we see, those 2 genomes are different, but the call is highly correlated. And the same thing holds true across all the 6 different comparisons. So now we're able to merge the TE presence absence call into one non-redundant TE frequency set. So now we have the matrix, we're able to answer a few questions. We can start with the frequency of the TE, what is it in the panel? We could also answer the question, what's the relationship between the TE similarity, which is a proxy of age, to its population, to its frequency in the population. And also for a nested TE element, what is the relationship between the nested and its outer element? And finally, which I think matters more to breeder for genome-wide association study, is to see how many Ts are not tagged by nearby SNPs in this population. To start with, we look at the frequency of the T in this panel. So T, it's been defined into different orders based on how they are replicated in the genome. Because the large numbers we are only focusing on the LTR, TAR, and Helitron, those 3 TE types, TE orders. So here comes trivia too. Out of the 445,000 Ts, how many of T are fixed in the population, just like the core genes? So you can think about the percentage, and I move forward quickly because we're getting close to the end of the seminar. So when we look at the frequency distribution of all those non-redundant Ts, we can start it from here, the TAR, Helitron, and here's the TAR. We can see that there's only around 6% of the T that's fixed in this population, suggesting there's a large number of T that's still segregating, possibly contributing to the different phenotypes we're seeing. We just don't know T is the causing variant. The second question where we could look at is the frequency, the T age, and also its population frequency. So to date the age of the T, we look at the 5' and 3' of the T sequences, where we can use the sequence divergence to get an idea of the age of insertion. And also the similarity of the LTR sequence within the insertion could be used as a proxy for the age. So here's what we're saying. We see the age is negatively correlated with its frequency in the population. When we break it down into different frequency class, we can see there's lots of young T, very young T, that's in low to moderate frequency in the in the population. As we know, T tend to create new phenotypes, especially those young T members will be so important to look into. So let's say where they are in the genome. So for the all LTR we are looking at, it's pretty much equally distributed across the genome, where we can see for the very young T that's at moderate or low frequency, they are enriched in high recombination region. And for breeding and genetics improvement, I think there's a good chance we're able to make crosses to make some sorts of different phenotypes. On the other hand, we also see there are lots of T with high sequence identity, but it's also fixed in this population. Same thing, we want to look at where they are in the genome. So all LTR equally distributed, all fixed LTR, you can see the trend start to shift where it's heavily biased towards the low recombination region. And for the high similarity T and at fixed in the population, it's enriched in the low recombination region of the genome. So the next question we look at is the nested element. What is the relationship between the nested outer element? As for the classic Brown's locus, we can see T likes to insert into each other, which is quite challenging to study. So we wonder what is the frequency just between those nested inner elements or its outer element if one of them is significantly higher than the other one. So what we can see here on the x-axis where we have those nested T and we have the outer element on the y-axis, what we could see is that the nested T element is less frequent than its outer element. Further, we wonder which one goes in first, which one goes in second, or something else has happened for the nested element. Here, we're able to break down the question into a plot. So we would think for the outer element, that's more frequent in the population, and it should be older than the nested element if they insert later, if the nested element inserted later. So let's take a look to see if data is supporting that. So after we visualize all the data point, the nested pairs, we're able to see that outer element is more frequent and older than the nested element, which was in some way validate our hypothesis where the nested element goes in later. But we also see that for such a small frequency differences window, we see there's a dramatic age differences. So that leads us to more questions than answers, whether those nested elements insert at the same time, but they accumulate mutations at a different rate, or it's like inserted at different times, but they just copy and paste and eventually get to the same pace in this population. So this question will be further answered in the future work. So last part I want to talk about is how many T's that's not been tagged by nearby SNPs. So when we look at the highest observed LD between a SNP and a nearby T within 1 million base pair window, we can see there's a large amount of T that is in the high LD with a SNP. So the future work use the SNPs, we're able to capture a signal if there's a T causing the phenotype differences. However, we do also see there are lots of T that's in low LD with a SNP, possibly not being paying attention, or what I should say, we don't have the power to detect them from before. So breaking them down into different T orders, we see majority of the Ts are in the high or moderate population frequency, not population frequency, high or moderate LD with the nearby SNPs, and the ends, the dark blue color that's the part we're gonna focus more in the future where it hasn't really been thoroughly looked at. So to summarize all the talk, we talk about the gene content variation, where it could contribute to a lot of phenotype we see, but we don't know why. And also we look at the genes diversity, where we know the fact that we need more gene to truly capture the diversity of maize. From the TE part, we now know there are lots of transposable elements, so-called dark matters, is actually actively segregating in the population and driving the phenotype differences. And also there are lots of TEs that we can look further that's not being tagged by nearby SNPs for the genome-wide association study in the future. At the end, I want to wrap my talk by saying that Maize is a model system where we can pave the road for lots of work for the future. I come from a minor understudy crop background, and I think what we're doing in Maize could totally benefit the understudy crop breeding genetics work. Last but not least, here I like to acknowledge lots of people. Candy Hirsch group here. Those people are so great to work with. In Nathan Springer's group, we work on TE a lot. In Iowa State, Matthew Hoffer's lab, where we work together on the Maize name Pan-Genomes. And also we all did a review manuscript here. And the whole name group, without them, the genome assembly could not be possible. It's like 4 or 5 years of work. Also, Minnesota Supercomputing Institute, where provide the high performance clusters. And I also want to thank the CFANs for providing me the postdoc fellowship. And all my talk content is included in these 3 manuscripts I'm showing here. So today is actually the New Year in Chinese calendar. So I wish everyone a happy Chinese New Year if you're celebrating. And I'd like to take any questions for now. Thank you for your attention and stick around.
So are there any questions for Yinjie?